Over the next several videos, we're going to start exploring the lipid bilayer and how fat tissue is developed. The central molecule to lipid bilayers as well as fat tissue are fatty acids. Fatty acids have two main features. One is the acid functional group, which is a carboxylic acid. We've learned that our carboxylic acid functional group looks like this, right? So we have a carbonyl attached to a hydroxide or to an alcohol group. So this is our carboxylic acid functional group. This is the acid part of the fatty acid. And the fatty part means that it's a nice long carbon chain. Okay, so we have a long carbon chain attached to this. All right? So this is really kind of the core of a fatty acid. We have a carbon chain that contains just carbons and hydrogens attached to a carboxylic acid head group. Okay, this is the structure of a fatty acid. So in this video, what we're going to focus mostly on is figuring out how we can name these fatty acids. Okay, so I'll start off by mentioning that in biology, in your body, there are some common lengths of fatty acids. Okay, so carbon chain lengths of 16, 18, and 20 tend to be pretty common. We can definitely go larger or smaller depending upon what animal we're talking about or what bacteria we're talking about, but this range of 16 to 18 to 20 tends to be pretty common. Okay, it's very, very rare to have an odd number of carbons in the chain length. So it would be very rare to find 17 carbons in your carbon chain. Okay, so let's look at a simple structure of a 16 carbon fatty acid. Okay, so the 16 carbons means that there's 16 carbons in this fatty tail. So let's start here. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So 16 carbons. And our carboxylic acid head group has to be on the very last carbon. So I have my carbonyl and my alcohol. So here's the carboxylic acid head group. So this structure we just drew is a 16 carbon fatty acid. And naming fatty acids is pretty simple as well, especially when they're saturated, right? Meaning that they only have single bonds. So a saturated 16 carbon fatty acid we name by putting 16, which is the number of carbons, colon 0. And 0 here means this is the number of double bonds. Okay, so we have a 16 carbon fatty acid with 0 double bonds. So if you were told to draw a fatty acid that's 16 colon 0, you would know to draw it with 16 carbons and no double bonds. Okay, to draw 20 colon 0, let's move down here. Alright, so if your task was to draw a fatty acid 20 colon 0, then we would draw a 20 carbon chain with a carboxylic acid on the first carbon, or the last carbon depending on how you look at it, with no double bonds. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20 with a carboxylic acid on this last carbon. So this is 20 colon 0. Okay, things start getting a little bit more complicated when we put in double bonds, but not a lot more complicated. So the one thing that I want to really stress here is that in biological systems, the double bonds are almost always in the cis orientation. Okay, so when we're drawing biological fatty acids, we always draw the structure so it's a cis double bond. So let's go ahead and try drawing one of these. Okay, it's going to be an 18 colon 1. And this 1 tells us there's one double bond in the fatty acid chain. And I'm going to go ahead and put this at the 9th carbon. Okay, and we'll talk about how we can imply this once we draw the structure. Okay, so I'm going to start drawing it. I have carbon 1, 2, 3, 4, 5, 6, 7, 8, 9. So I'm going to put my double bond in here. That's 10, 11, 12, 13, 14, 15, 16, 17, 18. Okay, and I'll put my carboxylic acid head group on this carbon. So note here that I drew this in the cis conformation. That's important because it's a biological fatty acid. And it's positioned on the 9th carbon. And we'll count it out here. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So it's positioned on the 9th carbon from this end. Okay? This is an important point here. When you count from the end of the carbon chain that does not have the carboxylic acid, we call this the omega carbon, and we give it a symbol of this omega, this Greek omega symbol. This is the omega carbon. Okay, so when the double bond starts on the 9th carbon from the omega end, we would call this an omega-9 fatty acid. And to name this fatty acid, it would be 18:1, which tells us we have one double bond on an 18 carbon chain, and we put N-9 or N-9. And this tells us that it starts on the 9th carbon from the omega carbon. Okay, so it starts on the 9th carbon from the omega carbon. This is how we would name this according to our more biological guidelines. This fatty acid would be 18:1 N-9. However, there's also another way to name this. Okay, and this is going to be the more chemical way of naming this. If we start numbering the carbons from the carboxylic acid, we see we have 1, 2, 3, 4, 5, 6, 7, 8. So in this case, we still start on the 9th carbon. And this is only going to be true when we're dealing with 18 carbon fatty acids. But so here we start on the 9th carbon. The fatty acid starts there. So to name this according to the chemical nomenclature, we would put 18:1, so 18 carbons, 1 double bond. We would then put cis, because this 1 double bond is cis, and we would say delta 9, meaning this double bond, so this means the double bond is at the 9th position. The big difference between the two different naming conventions is that for the chemical convention, we start numbering from the carboxylic acid. From the biological convention, we always number from the omega terminus. Okay, and that's a big deal. Don't forget that. The other big difference is that for the chemical naming, we have to very specifically state the double bond is cis. But in the biological naming scheme, we don't have to state that because biological fatty acids are cis configuration. Okay, so in the next video, we'll go ahead and reinforce these two naming conventions, and we'll explore fatty acids that have more than one double bond.